All right. Our next presenter is virtually joining us from Tamaki. She's coming from South Africa. Alana is a senior researcher who specializes in plant pathology and has worked for Manaki Whenua for over 2 years now. Alana may not use them during her presentation, but she's actually fluent in three languages. Quite impressive. So her presentation will discuss the decade-long journey towards importing a weed biocontrol agent for the invasive Chilean needlegrass.
I hope you can see my slides. I've shared the screen. Can you see it? Good. All right. Thank you.
Yep.
Cool. So good afternoon, everyone. Today I'm going to tell you a story. It's a decade-long journey, but started many years before. What I'm reporting, well, I'm the person who's basically in the sprint for this project. And it has been a work of patience, both for Manaki Whenua and a study in persistence with our Argentinian collaborator, Dr. Frida Anderson, who is based at CONICET in Bahia Blanca, Argentina. So basically, basically, you'll start to see the analogies for what I call a journey. So the first step started basically over 2 decades ago, when a biocontrol program was initiated against the serrated tussock as well as Chilean needlegrass. It was initiated by Australia and then very quickly piggybacked on by New Zealand. And this started the late 1990s. And the reason we looked at it in Argentina is that these Nassella species are native to South America. And the reason why this work was undertaken to look for biocontrol agents is that the Chilean needlegrass and serrated tussocks are both serious agricultural and environmental weeds, both in New Zealand, and it is a weed of national significance in Australia. In New Zealand, currently there is approximately 400,000 hectares that are infested by this weed, and you'll see on the map I can't know me and left and right. To the left, you'll see a number of white dots with black arrows pointing to where there are populations of Chilean needlegrass within New Zealand. However, with a ecoclimatic suitability model that was published by Graham Bordeaux and et al. In 2010, shows that if the climate were to change, this weed has the potential to invade up to 15 million hectares in New Zealand, which is almost over half of the land mass that is New Zealand. So over a 5-year period, Frida undertook field surveys in the native range, throughout the range of the plant, and that you can see through all the different sites here. Identified 3 main suspects. The first one was a rust fungus, Puccinia gramineella, was found to be too specific and was not work on this was not continued. The second was a second rust fungus, Puccinia nassella, and it was found to be not specific enough, just couldn't get enough inoculum in the field to actually continue with the trials. However, luckily for us, there was a third rust fungus, a Uromyces pencanus, which was thought to be the most promising agent, as it was thought to be easily mass-produced. You could harvest the spores or collect the spores really easily. It could be stored for long periods at -70, so it allowed you to bulk up and store the spores so you could do your testing. And then heavy field infections showed that the plants actually died back, and then there was a reduction in seed. So I'm just going to segue a little bit. So you might be asking yourself, what are rust fungi, and why do we at Manaki Whenua work with predominantly rust fungi and smut fungi? Well, what we call them obligate biotrophs, and it's quite a big word, but what it means is basically they cannot live without their host. So they are not fungi that we can culture on a medium. They have to be associated with their host, so they are true parasites. They do not kill their host, they just take sustenance from them. And 26 of the 36 plant pathogens that have been released as biocontrol agents worldwide are either rust or smut fungi. So, and the reason for this is that they have very high host specificity. They often have very many dry airborne spores that are spread readily through rain and wind, and are often self-spreading and can range from 1 meter to up to 50 kilometers from their initial release sites. Right, so going back to the actual story on this Chilean needlegrass. So what happened was with Frida, once she got the spores, the rust into culture and had an effective isolate, which was UP27, which is just Uromyces site 27, where the fungus was collected from. Once they went into the glasshouse and to greenhouse testing, they found that there was a mycoparasite identified, and I'll segue again after this slide. It wasn't prevalent in the field, so there was no indication that there would be a problem with the mycoparasite. However, under these ideal conditions in the glasshouse, and as you can see on the picture here, that those little yellow things are pustules or the spores. So you would need that to use to do testing, are covered by this white mycelial mass, and that is the mycoparasite. And it took a little bit of time, but Frida was able to eliminate the mycoparasite from the culture by storing the rust spores at -70 degrees, as well as the pathogen doesn't kill the rust spores, does kill the actual other fungus. So now my segue. So what is a mycoparasite? A mycoparasite is where you have a fungus which parasitizes another, and this is just the natural enemy of the plant's natural enemy. These interactions and mycoparasitic interactions are very common in nature, and they form part of the microbiota of plants. So whatever occurs on the leaf, whatever microbes are on the leaf, it forms part of that on the plants. However, there is an issue when it comes to weed biocontrol and the use of rust fungi or smut fungi or any other fungi, is that with these mycoparasites, you get variable establishment and effectiveness of these fungal biocontrol agents. And on the left, you can see just a range of issues that could potentially impact our biocontrol, and it could reduce the infection pressure in the native range. It could reduce your safety. So when we import the fungus into New Zealand, does it come with these mycoparasites, and how would we eradicate it, or how would the collaborator that we're working with in country of origin, how would they eliminate it? There's a reduction in the availability of inoculum, so you won't have enough inoculum to do any host range testing or to actually do any type of pathogenicity. So trying to see whether your plants are susceptible to this fungus. And sometimes when you bring them into the country and you've eradicated them from the fungus, from your culture, and you do a release with a clean culture, there are a large number of mycoparasites that occur in the new range of this pathogen. And would they then have reduced field efficacy if they are attacked by these mycoparasites? However, there is also a paper that has shown that there's increased agent fitness when you remove these mycoparasites from your agent. Okay, so back to the story. So then, once we'd identified it, we'd identified the potentially most effective agent. We'd eradicated or eliminated the mycoparasite. We had to then do host specificity testing, and this is one of the most important steps in weed biocontrol, to make sure that the species that you're importing and wanting to release does not go onto any of your native and or commercial species in country. So there are no native Nassella species that occur in New Zealand. The closest relatives to the Nassellas are the stipoid grasses within the stipe tribe. And in New Zealand, there are only 3 native taxa present and only 2 which are endemic. So a host range test list was set up, and at the end, a total of 65 plant species were tested, which unfortunately did not include the 2 endemic New Zealand species. However, there were up to 14 different wheat cultivars that were tested, and barley, and a number of commercially important species. And the weed within the fungus was found to be host specific, and pustules only developed on the target weeds in New Zealand, and to an extent too specific in the fact that they did not infect the Hawkesbury or Auckland biotypes of the Chilean needlegrass. And this could be due to the fact that we had very few, or that Frieda had very few plant accessions with which to do the testing. So once the host specificity testing showed that it was host specific enough, we then had to do what we call the necessary steps. So we applied for an application to import and release the fungus. However, this permit that we were given in 2011 expired, and the reason for this was because Argentina signed what we call the Nagoya Protocol, which looks at aims to create new incentives to conserve biodiversity and the sustainable use of the biodiversity components, and then to improve the role of biodiversity in sustainable development. However, what this did is they signed it and then had no idea how to go about actually implementing the Nagoya Protocol, and effectively this put the project on hold because there was no export permits given by the Argentinian government in a 10-year period. So that's where the decade-long wait comes in. In the interim, and then in 2017, we reapplied to the EPA for a new application or a renewed application. What happened then was we got the unforeseen step, was that during the period from 2011 to putting in a new application in 2017, that the Australians had requested that further host range tests be done, and the results of those became available. And what was found was that pustules started developing on 2 native Australian oyster stiper species. Neither of those species grows in New Zealand, but with an abundance of caution by Manaaki Whanua, we and the Department of Conservation requested that we further test the 3 native New Zealand taxa. So, and then we were thankfully given 2 time waivers by the EPA to actually complete this work. And we have permission to import and release until 2024, April 2024. So in September 2021, we finally got an export permit was approved. So we thought, oh, great, we're heading in the right direction, and this is where this step comes in. However, they only gave us 3 months to export the fungus. So Frieda was quickly bulking up the rust. She eventually was able to send it on the 23rd of December, and it was, we thought, a really early Christmas present. However, due to COVID, the delays with the the courier companies, it took 7 days to leave Argentina. It kind of did a short whirl and a stay in Tennessee, and then it spent a couple of days once it got into New Zealand at the FedEx facility due to a communications issue. And we received the spores on the 18th of January, so literally 3.5 weeks. And what this does is basically, so what it does is it basically has left the spores at who knows what temperatures for an extended period of time. And these are very fragile, spores are really fragile. And we rushed them into our containment facility here in Tamaki Makaurau, did the inoculation, and unfortunately, after a month, we saw no development of pustules. So that shipment, after 10 years and so much excitement, had failed. So once Frieda and her team had heard that it had failed, they then had to grow plants from seed again in Argentina. Thankfully, they had kept some of the fungus in the fridge at -70, and they were able to thankfully establish a new culture of the rust. And so the same culture that was used for the testing and everything throughout has remained the same. So we are using culture UP27, and we then had to restart the export process, and we thought that it might take as long as it did, and hopefully not the 10 years. but literally we were given, Frida started it, and we were given permission to export from Argentina within 2 weeks, which is fantastic. I then traveled to Argentina on the 15th of December. And the reason we did was that we could get a turnaround time of literally 48 hours. So I traveled and spent 5 days in Argentina. I spent time with Frida in her lab, and we harvested the spores, fresh spores, and literally within 48 hours, I had them in New Zealand, and I inoculated the New Zealand Chilean needlegrass. And what I hadn't realized at the same time was that I had imported my own fungus, and I came home with a bad dose of COVID, which I'd collected subsequently in Argentina. We did an inoculation, and I'm very happy to say that I can share the Whoohoo steps, which is that I've managed to establish the rust successfully in containment on the Marlborough accessions of the Chilean needlegrass, which is fantastic. So after 10 plus years, we finally have a living culture of this rust in containment. The steps forward is that I will need to complete the host range testing. We will then apply for an amended EPA permit to release, and then finally do the releases on the Chilean needlegrass in the Marlborough, Blind River Marlborough area. Basically, this is just the last step before the acknowledgements. This is the first, so I've undertaken one replicate of testing, and the rust only produced pustules on the actual Marlborough Chilean needlegrass accession. There was some infection, but there's a response by the plant which actually effectively stops the rust from producing the pustules on the Ostrostipes stipoides, which is shared. This grass is shared between New Zealand and Australia. It was tested, so I found out afterwards that it has been tested previously, and it gave similar results. On the 2 native species, which is the Achnatharum petrii and on the Anemoncele leesoniana, there was some infection, small chlorosis, but nothing on the Achnatharum petrii, which is fantastic. So, and I will be doing additional testing, repeating this twice again, just to confirm 100% that there is no, that the rust does not produce pustules on these grasses. And then there are those that make these steps happen. I'd like to thank our funders, the National Biocontrol Collective, and then the SIF funding, as well as our contributors from Argentina, as well as the Liam Falconer and Darren Underhill from the Marlborough Regional Council and Hawke's Bay Regional Council, respectively. Thank you very much.